Welcome to The Explainer. Today, we're going to completely shatter that classic high school textbook myth of the static, bean-shaped powerhouse. You know the one, right? For decades, we've all been shown these drawings of simple little ovals just sort of floating aimlessly in our cells. But the reality? Well, the reality is vastly more complex, incredibly dynamic, and absolutely vital to your survival.
So let's just dive right into this explainer. We're going to take a microscopic tour to discover how the mitochondria structure isn't just a basic container, but an active, shape-shifting machine. We're going to look at exactly how these tiny organelles are constantly rebuilding their own internal walls just to keep your body fueled. To truly understand how this works, here is our roadmap for today. We'll be looking at 4 main areas. 1. Gateway and barrier. 2. Membranes. 2. Secrets of the intermembrane and matrix. 3. Cristae, the living, breathing maze. And 4. Structure fails, function fails.
All right, jumping right into Section 1, Gateway and Barrier, where we look at the 2 membranes separating the mitochondria from the rest of the cell.
Think of the contrast here like a medieval castle. The outer membrane is like a highly permeable, bustling outer gate. It's packed with these VDAC channels, which allow fuel and small molecules to easily just flow right on in. But the inner membrane? Oh, that is a strictly guarded inner fortress wall that barely lets anything pass freely. It absolutely refuses to let ions just wander through, relying instead on highly specific transporters. And it literally has to be this strict. Because if that inner wall were leaky, the electrical charge required to make your body's energy would just wash right away.
Now this is wild. The outer membrane isn't just a passive wrapper. Proteins like Bax and BAC can actually open pores in this outer layer to release signals that trigger cell death. Inside the mitochondria, there's a molecule called cytochrome C, which is basically an essential worker for energy production. But if the outer membrane gets a signal that the cell is irreparably damaged, it opens up and spills that cytochrome C into the main cell body. It's literally a self-destruct sequence, triggering apoptosis. So the outer membrane is essentially playing God. It controls cellular life and death. Moving deeper inside, we hit Section 2. Secrets of the intermembrane and matrix, to uncover what lies between and beneath these walls. Now you might think the gap between those 2 walls is just empty space. But actually, scratch that. It is anything but empty. The intermembrane space is a highly specialized, active microcompartment. It basically acts as a staging ground where protons are heavily concentrated. This creates the intense chemical pressure that is absolutely essential to drive your cellular engines. Pushing past that inner membrane, we finally hit the matrix. This is the inner sanctum. It's a thick, bustling factory floor packed with enzymes, where raw materials from the food you eat are chemically processed. Specifically, the TCA cycle oxidizes fuel to harvest high-energy electrons. And get this, it's remarkably also the vault where the mitochondria keeps its very own separate genetic code. Let's see how all this brilliantly builds together in a sort of molecular relay race. First, the matrix prepares the fuel, handing electrons off to the inner membrane. Then, that inner membrane pumps protons to create an electrical gradient. Finally, that pressure drives the ATP synthase turbine. It is stunningly coordinated. The liquid matrix extracts the energy, the inner membrane builds the electrical pressure, and then those spinning ATP synthase turbines crank out ATP, which is the literal energy currency of your life. Okay, bringing us to Section 3, cristae, the living, breathing maze. This is by far the most deeply misunderstood part of the architecture. You see, cristae are not just passive wrinkles sitting there to increase surface area. If you look at older diagrams, they just look like random zigzags, right? But they are actually highly engineered microenvironments. They're actively organized platforms acting like specialized reaction chambers that manipulate the flow of protons on a highly localized level. The spatial division of labor here is absolute genius. The flat areas of these folds handle the electron transport, but the sharply curved edges are specifically shaped by ATP synthase pairs to maximize energy recovery. Let that sink in. The proteins physically dictate the shape of the membrane. The flat zones pump the protons out, and the extreme curves naturally funnel those exact protons right back into the ATP synthase machinery to maximize efficiency. But wait, how do they keep all those protons from just washing away? Well, to keep these microscopic environments stable, something called the MICOS complex steps in. It acts like a door frame, creating narrow bottlenecks. If the cristae folds were just wide open, the intense concentration of protons would easily dilute into the rest of the organelle. The MICOS complex literally solves this by pinching the entryways shut, creating isolated, high-pressure pockets that are absolutely perfect for generating power.
And this whole structure? It's completely alive thanks to dynamic architect proteins like OPA1. OPA1 continuously remodels the shape of the cristae depending on how much energy your cell needs at that exact moment. If you start sprinting or if a cell undergoes stress, OPA1 acts like a master builder, literally breaking down and rebuilding the internal walls of the cristae on the fly, just to optimize your energy output.
Holding all of these extreme curves together is cardiolipin. This is a special lipid inherited from ancient bacterial ancestors, and it perfectly stabilizes this complex machinery. While most cellular lipids have just 2 tails, cardiolipin has 4. This unique wedge-like shape allows it to act as microscopic scaffolding, stabilizing the intense curves of the inner membrane and holding those massive energy-producing protein complexes firmly in place.
Which brings us to our final stop, Section 4. Structure fails, function fails. We've seen all this brilliant architecture, but what exactly happens to human health when these microscopic structures collapse?
Well, the results are devastating. For instance, if that architect protein we talked about, OPA1, fails, the optic nerve starves, leading directly to vision loss. The optic nerve relies heavily on continuous energy to send visual signals to your brain, so a failure in OPA1 means the mitochondria lose their shape, the cell loses power, and vision degrades. Similarly, if cardiolipin is defective, it leads to something called Barth syndrome, which causes severe heart and muscle weakness. Without that proper 4-tailed scaffolding, energy-hungry heart muscles simply cannot develop normally.
This right here is why that old powerhouse of the cell or factory analogy kind of falls apart. A human-made factory cannot dynamically reshape its own walls or merge with the factory next door to survive. When a cell needs more power, mitochondria will actively fuse together into massive, connected networks to share resources. And when a section gets damaged, they just pinch it off and destroy it. They are fluid, living networks that are constantly adapting.
When disease strikes, it kicks off a terrifying, vicious cycle. Structural damage destroys energy production, which in turn causes even more of the architecture to collapse. A defect in the cristae lowers energy output, which increases toxic reactive oxygen species. That toxic environment then damages the very lipids and proteins holding the mitochondria together, which flattens the folds even more, dragging the entire system down into complete failure.
So, the ultimate takeaway from our explainer today is this. In the mitochondria, structure doesn't just hold the function. The structure is the function. The physical folds, the strict barriers, the incredibly specific placement of channels, these aren't just background details. The precise, shape-shifting architecture of this organelle is exactly what makes the chemistry of your life possible.
But all of this leaves us with a pretty fascinating mystery for our next exploration. Think about it. If this is really just an energy organelle, why does it have its own independent, ancient genetic code hiding deep inside the matrix? We've mapped out the physical structure today, but next time, we are going to dive into the genetic ghosts of the past that are still living inside your cells. Thanks for joining me on this explainer, and what do you think? Keep your curiosity fueled.